In this short video, I'm going to introduce you to the different types of blood vessels found in the cardiovascular system and their microscopic anatomy. There are three major categories of blood vessels: the arteries, the veins, and the smallest blood vessels, the capillaries. Arteries, by definition, carry blood away from the heart. A is for arteries and for away. Veins, by definition, carry blood towards the heart. To help you remember this, remember that in Latin, the word venet means to come. So in the veins, blood is coming towards the heart. The capillaries are the blood vessels that bring blood directly to the cells of your body. The capillaries form highly networked beds that allow for easy exchange of oxygen, carbon dioxide, and nutrients. Now I want to talk a little bit about the structure of the blood vessels in general. All vessels contain a lumen that blood passes through. The lumen is the empty space in the center of each blood vessel that, in living tissue, is filled with blood. So if I were to sketch out a blood vessel, this is my vessel wall, and this space here in the center is the lumen. The walls of both arteries and veins contain 3 layers, and these layers are sometimes called the tunics. The innermost layer is the tunica intima. This is the epithelial layer that lines the blood vessel. These cells are simple squamous epithelial cells known as endothelial cells. In vessels larger than 1 millimeter in diameter, there's a thin layer of connective tissue just outside the tunica intima. Outside of the tunica intima is the tunica media, the middle layer. This layer is made up primarily of smooth muscle cells. These smooth muscle cells control vasoconstriction, that's making the blood vessels smaller, and vasodilation, making the blood vessels wider. When the smooth muscle cells contract, the lumen narrows, that's constriction. When the smooth muscle cells relax, the lumen widens, that's dilation. This layer also contains elastic fibers, which are particularly important in the larger arteries. The tunica externa is the external layer, the outermost layer of the blood vessel wall. This layer is thick connective tissue, full of collagen fibers, and is also sometimes called the tunica adventitia. You see lymphatic vessels and nerves running through the tunica externa as well. In larger vessels, you see a network of capillaries called the vasa vasorum. Large blood vessels are too thick to get their oxygen and nutrients from the blood that runs through the lumen, so the capillaries of the vasa vasorum bring oxygen and nutrients to the tunica media of the large blood vessels. To recap, in arteries and veins, we have a thin tunica intima surrounding the lumen. Around that is the tunica media, the muscular layer. The tunica media can be quite thin in the veins and quite thick in the larger arteries. Around the tunica media, you have the tunica externa, the surrounding connective tissue layer. This can also vary in thickness. Arteries and veins contain all 3 layers, but capillaries contain just a thin tunica intima. Capillaries also have smooth muscle-like cells surrounding them that are called pericytes. Pericytes help control the permeability of the capillaries. but they are not a complete muscular layer like the tunica media. This slide shows an overview of what the vessel walls look like in arteries, veins, and capillaries. We'll summarize these differences in a moment. For now, pause and take a look. Compare the arteries and the veins and see what differences you observe. Pay particular attention to the tunica media. This slide compares the arteries and veins histologically, what you would see under the microscope. You can see differences in the lumen of the veins and the lumen of the arteries. You can see differences in the tunica intima of arteries and veins, differences in the tunica media, and sometimes you can see differences in the tunica externa as well. We'll summarize these differences in just a moment. For now, take a good look at this image. When you're comparing larger arteries and veins under the microscope, with practice it can get pretty easy to tell the differences between the two. In arteries, the lumen tends to be rounded, but in veins, the lumen tends to be flattened and may look collapsed. This is also why the lumen tends to look wider in the veins. Veins have thinner vessel walls that don't hold their shape when prepared for histology. The tunica intima, when you're looking at arteries under the microscope, tends to look ruffled. This is because arteries have a very muscular vessel wall, and they're still somewhat contracted when fixed for histology. In veins, the tunica intima will look smooth, even. Arteries have a very thick tunica media, at least the larger arteries do. In veins, the tunica media is much thinner. Note, though, that if you're comparing a small artery to a bigger vein, these differences might not hold up. But if you're looking at arteries and veins of approximately the same size, the artery will have a thicker tunica media. Lastly, the tunica externa may be thicker in veins, but this can be pretty difficult to see under the microscope. Arteries and veins frequently travel bundled together, like you can see here. There's an artery, and there's a vein. Arteries and veins are also frequently bundled alongside nerves. This is your nerve over here. Oftentimes you'll see a lymphatic vessel bundled together with the blood vessels and the nerves. All of these conduits travel through the body together, wrapped up in connective tissue and padded with the adipose tissue you see here. We all know by now that the heart pumps blood through the cardiovascular system. That blood flows first through the arterial system, through large arteries, and then through arteries of decreasing size, before entering into the capillary beds, and then returning to the heart through the veins. After coming out of the capillary beds, blood passes into small veins called venules, and these venules drain into slightly larger veins, which drain into larger veins until ultimately emptying into the heart itself. Don't worry about those green structures for the moment. We'll talk about those when we get to the lymphatic system. After studying the vessel structures, you should be able to answer these 3 recap questions. Go ahead and try it now, from memory. For the first one, think about whether they're carrying oxygenated or deoxygenated blood, and think about what type of gas exchange is happening in the capillaries in the lungs versus the capillaries in the other tissues of the body. For question 3, think about what happens at the capillary bed, and ask yourself this: will you get better gas exchange through a brick wall or through a sheet of paper?